Okay, start over. Okay, so today we're going to be covering the first part of chapter 14, which is vaccines. And then next time we meet, we'll be talking about how we can use things like antibodies, enzymes that are used for replicating DNA, that kind of thing, for diagnosing diseases and different things like that. Okay. All right. So here is what we're going to be doing today. We're going to be going over the learning objectives. I'm going to open things up for questions. We're going to talk about the goal of vaccination. Then we're going to talk about the types of vaccines and how they work. And then we'll talk about any concerns about vaccines that you have. Then we'll talk about what to do for next time. All right, so this chapter helps us move toward being able to meet the following learning objectives. Being able to define the key role of evolution as it applies to microbiology, identify microbial structures and connect the structures to their functions, identify key agents and processes of microbial information transfer, identify pivotal components of microbial systems important to human health, and analyze and describe the impact of microorganisms. Okay, any questions? Any questions to start? Hey, if you have any questions, let me know. I'm happy to answer them. Okay, so let's go to the whiteboard and we will talk about the goal of vaccination. Slide my chair here for it. Okay. Okay, in the normal course of events, and this is, pre-vaccination days, what would happen, and this ties into what we talked about in the previous 2 weeks when we talked about immunity. Okay? So I become infected with a pathogen, okay, it gets in, it's doing the incubation, it's doing attachment, it's doing colonization and invasion, and then we start to get disease symptoms, okay? Now, your innate immune system recognizes that there's something foreign right away, okay? And it's chugging along doing the best that it can. But if you develop a disease, then the pathogen has been able to get around your innate immune system. Okay? Now, meanwhile, your adaptive immune system has also kicked in. Okay? But as you know, it takes about between 3 to 5 days for your adaptive immune system to even get going. Okay, and it can take up to 14 days before you get protective. Okay, so the plan would be, actually, let me get rid of this because I'd like to draw it at a slightly different slope. So we start to see it 3 to 5 days. It ramps up, you get protection. Okay, and in the process, we get rid of the pathogen. Okay. And at least that's what happens unless, you die first, which is kind of a grim reality. Okay? And then if I am exposed later to the same pathogen, okay, then once again, your adaptive immune system recognizes things right away, it's fighting. If it's not able to do that, your adaptive immune system kicks in right away. We have a much better response. And oftentimes we can shut down the infection before you even know that you've got it. Okay, so before you have disease. So we'll say this is like a second exposure, a year later. And So that's why, depending upon the pathogen tends to change its antigens a lot. Then we don't see this. But generally, if you get ill with something and you survive it, then you tend to not become sick with it again. Okay. And that's what happens with a natural infection. Okay. And last week, we didn't get a chance to talk about the types of immunity, but what I'm describing here would be natural, okay, which is being infected with a pathogen. Active, okay, active because I've made memory cells. Okay, immunity. Now, the goal of vaccination is to replicate this, but without you getting sick the first time. Okay. Because, you know, there's always the chance that you're not going to survive, okay, the first time around. So what we do is instead of exposing you to a pathogen, okay, what we do is we come in and we expose you to parts of a pathogen. Okay. So the goal is to get your adaptive immune system ramped up. Okay. And oftentimes we'll come in with a second exposure to ramp up your adaptive immune system even more. Okay, so that we're already up here so that when you're exposed to the pathogen, then you don't get sick. Okay, so the goal of vaccination is to give you this first exposure. Okay, to antigens. So that when you have a second exposure, okay, which this time is to the actual pathogen, okay, the goal is that you not get sick. Okay, and the goal is to reduce illness, death, secondary complications to being exposed to a pathogen. We call this the post-illness sequela. Okay. Prime example is long COVID. Okay, and some of the problems that people have been having who, you know, have been, you know, have gotten ill with this virus, and if you become ill with influenza, it can leave you open to becoming ill with other respiratory diseases, because influenza goes and kills your ciliated cells that move the mucus up and out. So it makes you more susceptible to, say, bacterial pneumonia, okay, which would be a secondary infection. So we are trying to prevent secondary illness. Okay. All right, any questions about the goal of vaccination? Oh, and I forgot one other thing. Not everybody can be vaccinated, and we'll talk more about that when we talk about the types of vaccination, that the different types of vaccines. Okay? But if we get enough people surrounding the folks who can't be vaccinated, then we develop herd immunity. Okay? And all of that goes back to this. Okay, okay, any questions?
So the red line is how many antibodies? Or
the red line is more the pathogen, the numbers of pathogens.
okay.
Yeah, so that would be numbers of the pathogen. Okay, as you know, as it gets in, its whole point is to multiply. And then the brown would be the action of your innate immune system.
Okay.
Yeah, and then the orange, that would be your adaptive. And then the green would be your vaccine. Yeah. Okay, other questions? Okay, let's move on to the different types of vaccines. Okay, and when we talk about the different types of vaccines, we'll talk about what some of the good things about them and some of the bad things about them, because nothing is perfect. Okay, so we're going to start out with what are called we'll start out with attenuated. Okay, attenuated is a fancy dancy term for weakened. We also call them live vaccines. Okay, even when we're talking about a virus, virus is not alive. Okay, so it can't be a part of a live vaccine. But because we're using pathogens that have been weakened, okay, they are able to replicate, okay, or if we're talking cellular pathogens, grow in the patient, okay, and so that's why we call them live vaccines, because they can get in and do some of what a pathogen does, but by weakening it, we have removed most of the virulence factors. Okay, so they are avirulent. Okay, that is the goal, is to have an avirulent strain that will stimulate a strong immune response.
How do they make them like that?
Good question. So there's a couple of ways that we can weaken a pathogen. Okay? One of them is in looking at the history of vaccination. There was a technique that was started in China. At least they have the oldest written record of doing it.
With the smallpox?
Yeah, with the smallpox variolation. And what they did is they picked a mild strain. So somebody who had, just got a few pox, a few pus-filled blisters, okay? And so they would pick the scabs from that and then grind it up. So one, you're picking a strain that's already avirulent. It's had, well, less virulent, okay, less, that has had mutations, so it's lost a lot of its virulence factors.
So like the same with the cowpox thing then that they did over in England or wherever it was.
Exactly, yeah, it was in England, yeah. So we're picking mild strains, okay, that have already lost through mutation a lot of their virulence factors. And then also by drying out the scabs, you're physically inactivating an awful lot, okay, of the virus. Okay, so we have physical inactivation. Okay, because going back to chapter 7, the last part of chapter 7, a lot of pathogens don't like being dried out. It'll kill most of them, but because you have bioburden surrounding them, they're still, to a certain extent, you're still going to have a few that have survived. Okay, so by physical inactivation, we are lowering the numbers. Okay, of the pathogen. Okay, and so oftentimes when we are doing a live vaccine, we try to keep the numbers below the infectious dose. So in other words, you've got fewer, it takes longer for them to replicate. Okay. And so that, enables your immune system to have time to come in and clear it out before you get disease. Okay. And then, and also with the cowpox, with vaccination, we are taking a pathogen and introducing it into the wrong host. Cowpox is adapted to infecting cows. So when it jumps to humans, it's just not really very good at it yet. Okay. And so when we do attenuation in the lab, we take advantage of this. Okay, so we'll take a human pathogen that's really quite well adapted to humans, and we will put them in, say, lab rats or mice or guinea pigs. Actually, I think there's I'm not quite sure how to spell guinea. It's not one of those that are interesting to spell. So we get it adapted to these guys. We pass it from rat to rat, from mouse to mouse, and it readapts. Okay, and then when we inject it into humans, it has lost a lot of the adaptions that it had as a human pathogen, and we don't give them time to readapt back.
So you kind of like go backwards with it, basically.
Mm-hmm. Yeah, so with a lot of our attenuated strains that we use for vaccines, a lot of them have been, we put them through either lab animals, or we'll put them in cell culture. So we can take human cells that have come from humans, and but they're growing in a flask. There's no immune system. And if you grow the virus for long enough, it's kind of like you've taken somebody who has won the championship in mixed martial arts. Okay, they're a good fighter, they're in shape. Okay, and then you take them to the Riviera and you leave them there for a year and say, eat what you want, drink what you want, lay on the beach, don't train. And then when they come back into the ring, they're gonna get beat up. They're not going to do very well. And so that's kind of what we've done here is with the pathogens that you grow in cell culture, if they have a mutation where they've lost a virulence factor, there's nothing that eliminates those mutations from the population. So the population keeps getting weaker and weaker as you get mutations build up and you don't eliminate them. And so that's another way to weaken the pathogen and help it lose its virulence factors. Yes, Mitchell.
So for I'm just going to use the flu as an example. So if someone got the flu, they'd put it on a culture and they just culture it and reculture it. And each time they do it, it'll get weaker and weaker. Mm-hmm. Okay, cool.
Yeah, in fact, we do that. We, because we do make live influenza strains or live influenza vaccines. And that's the one where you hold your head back and they dribble the fluid into your nose. Yeah, that's an attenuated influenza strain. What they do is they grow it in fertilized chicken eggs. And so once again, it's it's a host that'll replicate in. But you do it in chicken eggs, they don't have much of an immune system. And so yeah, that pretty well does it. Yep. Yeah, so we've done that with a number of pathogens. We did that originally with smallpox. And we still keep the cowpox virus around just in case. We've eliminated smallpox from the population at large, but you just never know. We've done that with polio. What we'd do is we'd take fluid that had infective, attenuated polio virus, put it on a sugar cube. Much better received by children than giving them a shot in the arm. Influenza, we still do influenza. But the problem with, well, let's go through the things that are good. Okay, so things that are good. Okay. You're exposing the patient to all, I'm going to put that in quotes, of the antigens. Okay. So you are getting billions and trillions, I'm guessing on the number, but a whole lot of B cells and T cells activated. And they're all going after different epitopes. And so it's harder for the wild pathogen to mutate and become resistant or to make the vaccine less effective, okay? Because you've got all of these antibodies, you've got all of these cytotoxic T cells, you've got all of these T helper cells that recognize a whole slew of antigens. Okay. Also, because it is multiplying, okay, you've got plenty of time for your immune system to recognize there's a problem and respond to it. Okay. So we get really good immune responses to attenuated vaccines. Okay. But there are some problems. If you are immunocompromised, if we give you an attenuated vaccine, there is the possibility that it could give folks who are immunocompromised the actual disease because their immune system is suppressed. They don't have a very good immune system. And so these guys that are pretty much virulent become an opportunistic pathogen, and they can, it can be very bad for immunocompromised folks. So we don't want to give anybody who's immunocompromised an attenuated vaccine. You also don't want to give it to anybody who is living in the household. No, I'm starting out one way and change whatever, right? Start out with relatives. Because if you have a live vaccine that you've been given, they're going to be multiplying. You're going to be shedding a little bit of the pathogen. If you're in the same household with somebody that's, say, receiving chemotherapy or has received an immunosuppressive because they have rheumatoid arthritis or Crohn's disease, or they've had a kidney transplant, or you have an itty-bitty little baby that their immune system is still developing, yeah, you don't want to give them an attenuated vaccine. Yeah. The other thing is that there have been cases where the pathogen has mutated back to virulent. Okay, this process of attenuation can go the other direction. Okay, if it doesn't happen very often, but we did have a few cases of the polio vaccine reverting back to wild type, back to the virulent strain, and actually causing polio. Okay, it was less than 1%. Okay, so it didn't happen very often, but it is a risk. And it is a risk that we take in using attenuated vaccines. So any questions about attenuated?
So even if they're weakened, they can get back to being stronger.
Mm-hmm.
Okay.
Yep, because it's all about the mutations. You can have a mutation where you lose a gene, but you can have another mutation that restores the gene. Yeah, it doesn't happen very often, but it could. It could happen. Okay, are we ready to move on to inactive? Let's move to.
What would be the best kind to give somebody who is immunocompromised out of all the vaccines?
Generally we go with inactive. And then anybody in their household, we would also give them an inactive or killed vaccine. Yeah. And it depends upon the status of their immune system. Because like I said, some people should not be vaccinated. Some people have allergic reactions to vaccines, or they can have neurological problems that pop up. They don't show up very often. But for folks that have had adverse reactions to vaccines, we generally say, yeah, don't get that particular vaccine. And if you're immunocompromised, sometimes we say, hey, your immune system is at a point where you would not develop immunity if we gave you a vaccine. And so we're going to plan on everybody around you getting vaccinated to protect you from the pathogen instead. So that's generally what we recommend. And once again, like I said, it depends upon the specific clinical situation of the patient. But we have different types of inactive or killed vaccines. And the first one is what we call a whole agent. And that's where we take the entire pathogen and we kill it. Okay? And there's a number of different ways to do that. Okay? One of them is to explain, and this goes back to chapter 7, okay? Ways to control pathogens. Okay? One of them is to expose them to formalin. Okay, formalin is a dilute solution of formaldehyde. Formaldehyde is in the aldehyde family. And what that does is that cross-links the proteins. It freezes. Okay. The pathogen. And so since it can't move, if it's a cell, it can't live. But if it's, say, a virus, it can't do its job. Okay, it can't grab a hold of the host cell. Okay, but it keeps the proper shape. Of the antigens. So when you expose your immune system to it, then the antibodies that are produced are actually effective. Okay. We can do a similar thing with gamma radiation. Okay, we send that ionizing radiation into a bunch of the pathogen. We're knocking electrons off here, there, everywhere. And pretty much we maintain the proper shape. Okay. But there is a chance that you might get some denaturing. Okay, but we're pretty much, preserve the shape. Now, for some pathogens, okay, where they are heat stable to a certain extent, okay, we can use heat. Actually, I'm going to change colors. We're going to do red. The problem is heat only works if the antigen will go back to its normal shape. Okay, there are some pathogens that have heat stable antigens, so they will denature and the pathogen will be inactivated, they'll die. But then when things cool off, then those proteins go back to the proper shape. We inject them into people and they get an appropriate immune response. What can happen is that you can get denatured antigens that are the wrong shape. And so what happens is you do have a good immune response. It's just not going to work against the actual live pathogen. So we tend to not use heat very much. So some of the good things. Okay. We don't have to worry about mutations back. Back to virulent. We also use the term wild type. And that just refers to the most common type or strain that we find from clinical cases, okay, out in the wild versus in the lab. Okay? So we don't have to worry about mutations back to a virulent strain. Okay? We're still getting an immune response to all, I'm putting that in quotes, of the antigens. Okay. So once again, you know, even if there are mutations happening out in the world, out in the wild, you still are going to have lots of different types of antibodies, lots of different cytotoxic T lymphocytes going around looking for different antigens. And, you know, the pathogen's not going to change all of its antigens all at the same time. Okay. So we still get a pretty good immune response. Okay. Some of the drawbacks, because it's not replicating inside the patient, there's the immune response is less than for attenuated. So we tend to see lower antibody levels, okay, which is generally how we gauge how well a vaccination is working. Okay. So we are more likely to need boosters. Okay. Okay, so we can get that really good antibody response. Okay. But we can use this with some immunocompromised patients. Okay. Okay, questions about the whole agent inactivated?
So gamma radiation just kills it and it doesn't change its shape or anything?
It changes it less than heat. And it's a better option because some people are really allergic to formalin and it's hard to get all the formalin out.
Okay, so
So
gamma
it's
radiation.
kind of in between.
So it kind of changes it, but not enough to where your antibodies can't recognize it.
Yeah, for most of them, yeah.
Okay, cool.
Yeah, for most of them. Yeah, the whole thing is a balancing act. Let's see, was there anything else that's been mentioned? Nope, I think that's it. So I'm going to put inactive up here. And I'm going to put whole agent. Okay, the next one down we call a subunit. And we have different kinds of subunit vaccines. Okay, we've got some where instead of injecting the patient with everything that's in the pathogen, okay, we pick specific antigens. Okay, so like back in the day, well, let's go with purified. There's two kinds. There's purified and there's recombinant. So back in the day, what we would do for hepatitis B, okay, because that is not a virus we can grow in cell culture. We've tried, it just won't do it. So what we would do is, well, two things. We would either inject chimpanzees and then they would grow lots of the virus and we'd get their blood, pull the virus out and then inactivate it and then pull out specific antigens. Okay, so that would be purified. We'd also get people who had, who were carriers for hepatitis B. And so, you know, we'd pay them to donate their blood and we'd do the same thing. We would pull the virus out of there, inactivate it, pull out specific antigens. And in the case of hepatitis B, we were looking for spikes, okay? Generally speaking, we're looking for, in viruses, we're looking for spikes. Okay, in bacteria, we're looking for things like fimbriae. Okay, and other things, okay, that if we cover up those attachment structures with an antibody, then we're going to prevent infection. Okay, so that's generally what we're after. Well, there became problems with getting blood from humans, okay, because you just, you know, we know that they have hepatitis B, but there may be other pathogens that they have that even they don't know about. Okay? So what we've, and then also there's the ethics of working with chimpanzees in a medical situation. They're pretty close to our level of intelligence, and you know, they don't do well. You know, once we would infect them with hepatitis B, we didn't want the rest of their group to get hepatitis B. It's very highly transmissible. And so we would have to isolate them, and it's kind of like when prisoners are put in solitary. The poor chimpanzees would go insane. It's not a very nice thing. So we've moved away from doing that. So what we do is we take the gene for the antigen that we're looking for, and then we put that into a bacterium such as E. coli or a yeast. Okay. And what we do is we get them to make the antigen. Okay. So we're taking the gene from one pathogen, putting it into an avirulent bacterium or yeast that grows very well in the lab, and we get them to make lots and lots of antigen. Okay. And then we're doing just one kind of antigen. Okay. We don't have to worry about the ethics of using human donors or highly intelligent animals. And so, yeah, most of our subunit vaccines are now recombinant, okay? Which is a good thing because that's what we did with COVID. We can't grow COVID, the virus that causes COVID, SARS-CoV-2, we cannot grow it in the lab. But we have sequenced the entire genome. We found the spot where the spikes are. We take that part out, just the spike information from the RNA, use reverse transcriptase to make it into DNA. We put it into E. coli and we make all sorts of COVID spikes. Cool, right? And pretty much they did that when they were studying, you know, the SARS family, working on a vaccine. So we actually, the vaccine for COVID is not recombinant, but we used recombinant technology to get us toward making the vaccine. So things that are good, okay? Things that are good. Yeah, there's even less risk with using a recombinant vaccine, okay? Because you're not dealing with the entire pathogen. So there's lower risk. And then once you get to recombinant, we're not growing large amounts of the pathogen. And so there's less chance of accidents in the factory, people getting sick working in a vaccine factory. And so you're not growing the pathogen. And so it's safer. It's safer for everybody concerned. Okay. And once again, it's good for using on immunocompromised folks. And then also, it's a good way of developing a vaccine for a pathogen we can't grow in the lab. And we call that being unculturable. Okay. Now, part of the problems, okay, we get less of an immune response. Than we see from a whole agent inactive, okay? Because it's not replicating in the patient's body, okay? And we have fewer antigens that your immune system is responding to, okay? And so if the wild type pathogen mutates, then we might be making antibodies that are ineffective. And we have to come up with another version of the vaccine, which, by the way, is what we do with influenza. Yay. So part of that is because it's a whole agent inactivated, and part of it is just because influenza is annoying and it goes through some major changes on a routine basis, just because it's a segmented RNA virus. Okay. All right, questions about the subunit? Okay, rather than clearing the entire board, I'm just going to clear this part so I can leave my headers up. See how fast I can erase it.
So is subunit part of inactive?
It is part of inactive.
Okay.
Yep, it's considered a category of inactive because it's not going to replicate within the host. When you give a patient an injection, we don't have the live pathogen, and so we consider them all inactive or killed.
Okay, cool.
Yeah.
And then they target spikes in Fimbriae because with E. coli and yeast, those focus on spikes in Fimbriae.
We could put anything we wanted into the E. coli and the yeast, because they're going to make whatever proteins that we give them the genes for, as long as we set it up right. And then what we do is we pull out batches and we lyse the E. coli cells and then purify the proteins we're after. But we do this because they make what we call neutralizing antibodies.
So they make the proteins, but they don't make the spikes or Fimbriae.
The spikes in the Fimbriae are proteins.
Okay.
Yeah, because if I've got, or they can also be glycoproteins. Like if I've got COVID here.
Oh, I get it now. Okay, so they only make the spikes in Fimbriae, and so when the antibodies target it, they're only targeting the spikes in Fimbriae.
Right, and so they would
Okay.
not be targeting these little guys that also stick out, because we're looking for mainly, something that's going to prevent attachment.
Okay, cool. I had that backwards. That's
It's easy to do. Easy to do. Okay, now the book lists toxoid proteins next, but I'm going to go with conjugate next because it makes more sense. Okay, so I tell you what, let's get a gram-negative bacteria in here. And we've got some Fimbriae coming off. Okay. And it has two membranes. And this outer membrane has endotoxin, which is a lipid, L-lipopolysaccharide. Okay, and the actual toxic part is lipid A. Okay. Lipids, with the exception of lipid A, okay, do not make good antigens. They're just too floppy. They just kind of flop around, and it's really hard for an antigen presenting cell to present a specific shape for B cells and T cells to recognize. Okay. LPS does the exact opposite. It stimulates everybody indiscriminately, so it's equally bad. So you would not want to vaccinate somebody with lipid A. And then with bacteria that have capsules. Okay. Capsules are made out of carbs. And we have the same problem. They're kind of floppy. They have distinct shapes, but it's hard for an antigen presenting cell to present epitopes that are going to be recognized. Let's also take a look at another example of a capsule former where the capsule is a virulence factor. Okay. Streptococcus pneumoniae, the most common cause of bacterial pneumonia. The A virulent strain does not have a capsule. Okay. In fact, if we were to swab your nose or your throat, we could probably culture a virulent strep pneumoniae. Okay. But the kind that causes bacterial pneumonia, okay, has a capsule. This candy coating that prevents them from being easily phagocytized. Okay. And so when we were working on developing a vaccine, okay, we tried doing a attenuated vaccine where we had the A virulent strain, did not provide protection against the virulent strain. Okay. The problem was is we need to develop antibodies, okay, and cytotoxic T lymphocytes against the capsule, okay, against this specific virulence factor. But if we just take the capsule, because it's a carb, it's not really antigenic. So what we do is we do a conjugate vaccine. Okay? So what we do is we take a protein that we call a hapten. Or no, actually, it's called an adjuvant. Hapten is something different. And we stick the capsule carbohydrates on it. And so one, that makes the carbs a bigger mass for the antigen presenting cells to display. Okay. And then it's like they can grab a hold of this adjuvant, and then it's like all of a sudden they can see the carbohydrates around it. Okay. So we call this a conjugate, okay, vaccine. Sometimes we call them carbohydrate vaccines because we're stimulating the immune system against a carbohydrate. Okay. So we'll do that when there are virulence factors that are hard for your immune system to recognize. We will stick it to an adjuvant that just makes it easier for your immune system to see it and respond to it. Okay, questions about the conjugate? Yeah, so you're if when you get the pneumococcal vaccination, it's a conjugate vaccine. I think the Haemophilus influenza vaccine is also a conjugate vaccine. Don't quote me on that though. Okay, our last one, toxoid. That's where the goal is to develop antibodies against a toxin. Okay. So here's an example. Okay. Clostridium tetani, okay, is an endospore forming soil bacterium. We'll go ahead and put an endospore in there. We'll color it green. I know that kind of looks like a nucleus, but it's really an endospore. They live in the soil and they cannot grow in the presence of oxygen. Part of why they produce endospores. But let's say that I get a whole slew of these in my deep tissue, okay? So there's plenty of oxygen going to my cells, but it's carried there. So, you know, I step on a nail, I have a deep tissue wound, there's not a lot of free oxygen, they can start multiplying happy as little clams, okay? And one of the things that tetanus bacterium do is they release a toxin. And it is a neurotoxin. Okay? And so this neurotoxin causes your nerves to not stop their signals. So basically, as this toxin binds to your neurons, they continue to fire the signal to contract to your muscles. And so you get one big Charlie horse. And if you have the toxin in your face, your jaw clenches up so you can't open your mouth. And so we call it lockjaw. Tetanus, another name for tetanus is lockjaw. The problem is not necessarily the bacterium. Okay, the problem is the toxin. So what we do is we take the toxin, usually we inactivate it. Formalin has been very popular. Okay. And so that freezes the toxin in place so that it can't harm you, but we have the correct shape so that neutralizing antibodies can cover up that neurotoxin when you're first exposed to it, and it prevents tetanus, those cramps, okay? And then your immune system can wipe out the bacteria. Okay? So there's a number of diseases where the problem is not the bacterium, the problem is the toxin, okay? So the tetanus vaccine, that's one of them. It is a toxoid vaccine. We are injecting you with an inactive form of the toxin, okay, so that you can develop antibodies against it. Also diphtheria, okay? The problem is not necessarily diphtheria, it's the cytotoxin that it makes, okay? It kills a bunch of cells in your throat. You can get a pseudomembrane, it can start blocking off your breathing. It's not good, okay? So your DTaP vaccines are toxoid. Okay, questions? Okay, let's get to the newer ones real quick. I say newer, I'm gonna put it in quotes. Okay, newer, used in humans, okay? We have been using in veterinary medicine for decades, some vaccines that we used for the COVID vaccine, okay? So one of them is the mRNA, okay? Been used in veterinary medicine for many, many years to prevent, so far as I know, equine encephalitis virus disease. So encephalitis is inflammation of the brain. And you really do not want to get encephalitis, okay? And you don't want your horses getting it either. So we have that, and then we also have what's called a viral vector. Okay, and because I want you to know which ones are which, okay, going alphabetically, it's supposed to be no, Moderna and Pfizer are mRNA, Johnson & Johnson, and AstraZeneca. Actually, I should put AstraZeneca first. AstraZeneca, they are viral vector. I put AstraZeneca last because they haven't gotten approval in the United States. They have in Europe and Africa and Asia and some of these other places. So what happens is we take, okay, SARS-CoV-2 is an RNA virus, okay? And so what we do is we take just the sequence of the viral genome, okay, for the spike. Okay, so we take the spike genome, okay, and just for the ease of making more of it in the lab, we convert that to DNA. So then using the DNA, we transcribe that okay, into a whole bunch of mRNAs. And we take that and we inject that into the patient's cells. Okay, so human cells. And these cells, okay, they see an mRNA, they say, oh, gotta make it. And we make a bunch of spike proteins, and your immune system recognizes this and we stimulate the immune response. Okay? So it's kind of like a recombinant subunit vaccine, but we're getting our cells to make it. Okay. So then, you know, enzymes in your cells, they never leave mRNA sitting around. After the expiration date for the mRNA, they chop it up in your cells, stop making spike proteins. Okay. At least in theory. And now with the viral vector, it's very close to the same thing. We take just the spike genome. Okay, so we take the spike genome. We do the same thing that we do over here to make lots and lots of them. But what we do is we package it in a capsid. And then we inject this capsid into the patient, okay? And it's set up to grab a hold of the patient's cells and get itself in. So it, because the capsid does attachment, penetration, and uncoding on autopilot, so to speak, okay, it gets the mRNAs into the patient's cells. And we do the same thing as over here. mRNAs clamp onto it. They start making spike proteins, okay? And those spike proteins get embedded in the cell membrane, okay? It's just because we have packaged it in a capsid, you don't have to keep it frozen at those really, really cold temperatures that you have to for the mRNA. Okay, questions? Okay, we've got about 15 minutes left, so I'm going to skip asking you questions. Let's go ahead and move on to concerns about vaccines. Now, vaccines are not perfect, okay? There have been problems with them, and there are concerns about vaccines, okay? And they are legitimate concerns. But I figure in this segment, let's go ahead and talk about it. Let's address the elephant in the room. What are some of your concerns about vaccines? Or concerns that you've heard about in the media? Maybe you don't have those concerns, but you know, people are concerned about them.
I think one with COVID was some people were getting sick right after having the vaccine.
And that is a concern. Now, for most people, it's just, you know, you schedule that you get the vaccine on a Friday, so you have the weekend to recover and you're back to work on Monday. Okay, that is definitely a side effect. It's unpleasant. And I mean, nobody wants to say, okay, I know I need to schedule when I'm going to be sick. You know, it's kind of like scheduling surgery. But with some people, it lasted longer than 24 hours. Getting to the point, you know, where, you know, one of my friends, she had gotten COVID, a bad case of COVID. Then when the vaccines became available, when she got the first dose, of Moderna, she was sick for a week. It was like she got COVID again. And so she is not a candidate for further vaccinations, for boosters. She should not get them. Yeah. The sickness that she got was to the point where she should not be vaccinated. Yep. So definitely a concern, and that's why the manufacturers of the vaccines do their best to let you know what the potential side effects are. One, so you can watch out for them, and two, so that you know what the risks are. Hey, what are some other concerns?
Blood clots.
Blood clots. Now, we saw this more with the viral vector. And we saw it in a narrow range. It was mainly women in childbearing age who were on birth control and were on their fetal. And so birth control increases your chances of having blood clots. Okay. Being on your fetal increases your chances of having blood clots. Oddly enough, being inactive also increases your chances of having blood clots. It's better to have moderation. And so you add to that the vaccine, and we had some people dying from blood clots. Yeah. So now we know, we recommend that women in these categories should not get a viral vector vaccine. They shouldn't get the Johnson & Johnson vaccine. They shouldn't get the AstraZeneca. And if they do, they know to watch out for the symptoms and get to the emergency room immediately. And then we can give them some anti-clotting factors, and the treatment rate, the treatments have been highly effective. But it's a concern. It is a concern, especially if you are in childbearing years and you have these other predisposing factors. So it is a valid concern. Okay, what are some other concerns?
What about straight death?
Yeah. We have had some people die from vaccination. Part of this is it's really hard to tell unless you have an allergic reaction. That's part of why they have you hang out for 15 to 20 minutes after getting a vaccination. Because if you're going to have an anaphylactic shock, an overwhelming allergic reaction to it, it's going to happen quick. And so if you do have anaphylaxis from a vaccine, we can administer epinephrine, but that's not always effective. You can have an allergic reaction and you can die from it. We have had some people who have died relatively quickly after getting a vaccination who were healthy previous to that. Okay. And the problem is we don't have enough people dying that way, thankfully. I'm not saying that's a bad thing, but it means it's really hard to separate whether their death occurred from the vaccine or whether it happened from something else. Or if it's like with the blood clots where you're adding to undiagnosed predisposing factors. Yeah.
My aunt ended up with MRSA and SARS after her second one. Extremely healthy woman got the second one and died within a week.
Oh dear, I'm so sorry. Yeah, with the MRSA, I suspect that there was a problem with the injection.
Yep.
I am so sorry.
I'm
Yeah,
sorry,
so-
Devon, that's horrible.
Yeah. I lost three of my family members died that way.
Oh dear. Oh, that's horrible.
They thought they were doing their part.
Well, yeah. And that's one of those, it doesn't help for the family. Even if it is a very, very slight chance of it happening, when it happens to you and your family, it's still awful.
So
And it makes
how
you
would
think,
SARS
too.
kick in on that? Is it because the MRSA just wiped out the immunity?
Very well could have been. It could have been that the MRSA decreased the immune. And then when they ended up getting SARS-CoV, and it could have been SARS-CoV-1, which is SARS.
Yeah,
And or it could have been two. And so because they had MRSA, it decreased their immune ability to fight off the virus when they got it.
yeah, okay. Because any of those other viruses in the body then will weaken the immune, so then the other ones have the chance of taking off and doing what they're not supposed to do, correct?
Exactly.
And
Yeah.
that's why you're supposed to clean the site of the injection. So the parental, you don't get exposed from other problems from a dirty needle,
Right.
dirty
Right.
site.
And the problem is, and I don't know what happened. I wasn't there, but you can't sterilize the skin. And so, in these clinics, and in drugstores, getting the vaccination, you're more likely to have high numbers of MRSA just because you have more people coming through. And so you have to be more careful with your disinfection and your antisepsis. Yeah, so yeah, I'm so sorry. That's rough. And, if it were me, it would make me really nervous about getting a vaccine, especially at someplace, where they have this problem happen with my relatives. I would be reluctant. Change reluctant to almost phobic. So, yeah. Oh, yeah, I'm sorry about that. And unfortunately, this happens. There are a few people that do die after receiving a vaccine. Well, I'm going to pop up two more just real quick. One of them is neurological. There is a condition called Guillain-Barré syndrome. And we have no idea why, but there are some people that after they get a vaccination, and it doesn't matter which vaccination, they will have temporary paralysis. And it can be in a part of their body, or it can be a whole body response. And I'm not even going to attempt to spell Guillain-Barré. But it's usually it lasts from about 2 days to about 2 weeks. But there have been some cases that have been permanent. Okay. Some have gone up to a month, but usually on the first time, I'm sorry, 2 days of being paralyzed would scare the living daylights out of me. So with anybody who has gotten, and sometimes you can get Guillain-Barré for no apparent reason. But if you get it after getting a vaccination, you are not a candidate for further vaccinations. So that's why when you go in for your flu shot, they ask you if you had that.
Or story, one of my patients, after we gave him the flu shot, he got it.
Did he clear up
No,
quickly?
no, he still has some deficits from it.
I'm so sorry.
Yeah,
What's
it's
the
just when you
kid?
work in the public like that, you just never know.
You never know. And
You
you're
never
more
know.
likely in working in the public, you're more likely to see cases of that than of me actually personally getting it. But yeah, no, that's a risk that we take. And generally with all of these, we say, well, the risk of actually getting the disease is worse than these side effects. But these are definitely things to take into consideration. Last one, autism. I forget when. I'm gonna have to go back and look the date of the Wakefield paper again. There was a British physician that had done a study looking at 12 kids that had autism versus about a group of 24 control children. And he noticed two things in his report. By the way, I have the Wakefield report. It has been disapproved by the Lancet, which was the publication.
Didn't he also lose his medical license after all of that?
He did lose his medical license because he did some extremely unethical things. But I have the paper in case you want to read it. But one of his conclusions was that these 12 children also had gut issues. They had problems with constipation, acid reflux, digestive pain, okay? And he did not see the same things in the control children. They had healthy gut. And he did comment that with the children that had autism, it showed up at about the time that they were vaccinated. And so he suggested that mercury, which is a preservative that we use used to use in vaccines, we've since taken them out of all vaccines, should not be used for juvenile. Okay. And so he stressed this issue. Okay. And got a lot of people scared to death to vaccinate their kids. Well, it turns out he was working for a company that made a mercury-free juvenile vaccine. And then also he was working with patients, these patients who were suing the government. Okay,
Their parents were, right?
company.
They were already in an active lawsuit.
They were already in active lawsuit. And then for his controls, he had a party and for one of his kids, and he, without asking the parents or getting approval, said, "Hey, I'll give you $5 if you let me draw your blood." You don't do that with, 8 to 12-year-olds. You just don't do that. It's so unethical.
I'm guessing
So
that
anyway.
was the part that got his medical license removed.
Yes, as well he should. I mean, I'm laughing, but it was just, you just don't do that. You don't do that. You don't say, "Hey,
Not for $5, at least go for 20.
yeah,
That's what I was
20," and
about to
ask the
say.
parents, get the parents' approval.
I mean, come on, let's do a bidding war here.
Exactly. You want my blood? You're going to have to pay for it. So anyway, but he raised some valid concerns. So we've since done many, many studies looking at the connection between autism and vaccination. And we have bigger sample sizes. And we have found no difference in the number of cases of autism in kids that have been vaccinated versus those who have not been. We have the same rate of autism. What we have found, interestingly enough, resolving the gut issues, getting a healthy microbiome in the gut will decrease autism spectrum symptoms. It won't cure it, but it sure makes it less handicapping. Okay. Kids that were not able to speak were able to get to the point where they could speak, where they were able to communicate better. Still had sensory problems, but they were less. Okay. So we're thinking, the microbiome has something to do with this. We've also found that there's a higher incidence of children developing autism when the mother has had an illness that caused a high fever in the first trimester. And so there's something with, going back to the inflammation response, we're thinking there's something to do with the inflammation response having a negative effect on a fetus during those first crucial weeks of brain development. And then the other thing that we see is that symptoms tend to show up at about the time that kids get their second vaccination, but it's whether they get their second vaccination or not. There's also a genetic component. We tend to see autism running in families, whatever that means. So it's like cancer. You've got several predisposing factors that all combine to create this particular disease. But so far, we have not seen a connection between vaccination and the development of autism. So just kind of FYI. Okay, other questions? Questions and comments? Okay, that's pretty much it. We didn't get to the questions I was going to ask you, but I will put them in the practice quizzes. Any other questions before we end for today?
Do we have one exam or two left before the semester's over?
We have one. Yep, and it'll be available during week 16, so that including this week, 3 weeks from now.
Oh, that's nice. Okay, thank you.